to jump around a little bit. I'm going to skip some of the chemistry that's closer to the beginning, and we're going to just try to cover some other things today. So don't be surprised if we skip a few slides here coming up. I thought I'd put this one up here. People would ask me about my wife and dancing and that sort of thing. This was her very last performance. She danced professionally for about 30 years. And she was 45 when this shot was taken.
Wow.
She started my interest in anatomy. I forget which one she was doing. I don't know if that was Barbara. She was dancing with this Russian fellow. He actually came and lived with us for about 9 months, so that's where I picked up some Russian. I taught him English every night, and he taught me Russian every night. Got along pretty well. Anyway, getting into the nervous system. PBS here. I just wanted you to see where some of the things come from, though, that we talk about. This worm here, this is a nematode, just like the ones you saw in Lake Gilbert, crawling around in that algae. But there are hundreds of papers that come out on this guy every year about different sorts of things, the genetics, the structure, the stem cells, and how they use the transcription factors to differentiate between nerve cells and GI system and that sort of thing. A lot of what we know about what turns on things, we know because of this guy. They've mapped every single one of the cells in his body. We know where they came from, starting with the egg. And there's a little over 800 of them in the adult worm. Okay? And you can even buy T-shirts where the tree is all built up, so every single 800 cells is all around the T-shirt, and you can see where they came from. Okay? So that, but you have to know the transcription factors, what's going to turn on this cell and make it something else, and that sort of thing. And that's why they're so interested in this guy, because he's been mapped so well. We know the whole genome. We know every single gene and base pair in his system. In the same way with some of these other animals now. We now have a few hundred animals that have actually had their whole genome done. Okay? Remember, it was humans, the first time we had ours done was in 2001, and they were really going gung-ho on that for years and years and years. It used to take a long time to get certain sections to find the sequences. Now you can do it almost automated. And the cost is going to be coming down. Sooner or later, you're going to go in and get a blood test, and you're going to get your whole genome done just like you would a lab report. Okay? But it was a couple of $1 billion, the first one, the first human. It's now sitting around $1,000, I think, to get yours done. Okay? But it'll be down to a reasonable rate pretty soon once they automate it all. So, the squid, this guy, there was big research with him when I was in school, because it's the axon, the arm of the nerve cell that sends the message. Okay? The squid axon can be anywhere from 3 to 9 feet long, and it can be almost as big as a pencil lead. So that's a one cell that you're seeing, and that's where they could get the electrodes in there, and they could measure those electrical variations along the membrane and see the action potential travel along and figure out which ions were doing what. Okay, so a lot of research on what we just talked about for this upcoming test came from the giant squid. Okay, and then the fly. All his genetics are done, all the genes. We know how to turn a lot of them off and on. We know how to make freak flies. We know how to make flies with eyes on their feet and feet on their head. We know how to make flies with 2 or 3 sets of wings just by turning on different transcription factors and controlling genes to do that. Okay? So we're really learning a lot about what makes us us by studying these guys. Okay? So, and then you can throw some mice and rats in there too. They're pretty important as far as a lot of studies that have been done. But I thought that one might surprise you, the worm. Okay? I'll bet there's literally 100,000 papers that have been written on that guy. Well, this is the bad boy that we're going to be talking about for the next couple of weeks, the neurons. Okay? How they work, how they get together, how they function, and how they allow us to think. I'm not even sure this is human, but this is a typical neuron. You can see all these little projections sticking out here. One of them is going to be able to send that action potential out. Okay? All of the others, all these other arms and legs here, are bringing the potential in, okay? They're bringing a message in, okay? But every neuron only has one axon that will come out. Now that axon will sometimes diverge, but it's only one of those on. So everything, and you see all these little dots, okay, all this little grainy looking stuff? Every one of those is a synapse with another neuron, okay? It's estimated, well, our brain is estimated, we have about 100 billion neurons, okay? And every one of them, they think, averages about 5,000 connections with other neurons. Okay, so you're talking a very intricate, you multiply 100 billion by 5,000, that's a lot of connections, okay? A lot of synapses. It's a very complicated system. And this was one of the guys that actually came up with the stain. Remember Golgi apparatus? Well, this is your boy here. Okay, he was kicking around in his kitchen with his ammonia and silver stains, and he came up with something that would actually stain the neurons. And he started, looking at different parts of the brains. This looks like the hippocampus here. But he saw so many connections that he was just flabbergasted with the intricacy of it all. And then, like a typical scientist of his time, he said, well, I don't understand it, so nobody else probably will either. Okay. And they don't, when you hear those kind of proclamations, you just go, because a few years later, sure enough, we figure out how to figure out the connections, we figure out how the nerves, work, and that sort of thing. Like the guys that said, we'll never know what's in the sun, okay? And then a couple of years later, they invented the spectroscope, and they found all the elements that were in the sun. So that's the kind of thing to avoid if you're going to be a scientist. This is rat brain. This is actually using a virus. Okay? You can take the virus, will get inside the neuron, and then go down the dendrites or the axons and infect other neurons. Okay? But if you can put a virus in there that has a glow-in-the-dark gene, okay, a fluorescent gene, typically the one they come from corals and things like that, okay? You've heard about the glowing rabbits and the glowing tomatoes and that sort of thing. It's the same kind of thing, really. You put those fluorescent genes in there. But this virus now, you can see the pathway of the connections between this neuron and other neurons, okay? Because only the ones that get infected by that neuron are going to glow. Okay, and then you can see the pathway as it goes, wings its way through the whatever tract you're looking at. Okay, so that's just a typical sort of thing. Still, it's hard to tell which one of these is the axon. See, some of these dendrites are pretty big here. So that looks like a typical pyramidal cell in the brain, though. Now, most of this, there's not much on here really that's new for you. This is a graphic of a neuron. You already know the nucleus, the nucleoli, the endoplasmic reticulum, the Golgi, the mitochondria, the microtubules, and the little vacuoles in here, and other things like the lysosomes and peroxisomes. This is a typical cell, so it's going to have all that stuff. Okay, the microtubules are running down through the arms here. Remember, that's how the mitochondria is going to get dragged down into the dendrite to form ATP, so you can run the pumps and things down in here. Okay, so just like those little walkers, you're going to be taking that stuff right down those little arms and legs. The ones that bring signals to the neuron, okay, and I'll talk about the signal in a second here, but the information that's coming to the neuron, these are called dendrites. Okay, also all these are dendrites. The special one that sends a message out is called the axon. Okay, and there's a special area right here called the axon hillock. This area right here has to get positive enough. It has to get to threshold before you get the action potential fire down the axon. And it only fires in that direction. It won't go backwards. Okay, so this is where this voltage-gated sodium channel is. Okay, it's not a chemically gated channel here. It's voltage-gated. Okay, and now some of these out here are going to be chemically gated because they have transmitters, just like acetylcholine, that's going to be exciting the muscle. Okay, but this could be dopamine, this could be serotonin, it could be a dozen of any other molecules that the actual brain uses. Okay, so we're going way beyond acetylcholine in a much more complicated way. But this area, you're not going to get an action potential that runs along this membrane. Okay? It doesn't have those voltage-gated sodium channels out here that are going to give you an action potential. So what you have to do is build up this area of the cell up positive charge enough, okay, so that this will fire. Okay? So everything that's coming in, you're going to get positive charge around here, positive charge around here. Maybe if this is an inhibitory neuron, you might actually increase the negative charge and make it less likely to fire. So everything that's stimulating this nerve, whether it's excitatory or inhibitory, it adds them up like an adding machine. Okay? And when this nerve finally says, all right, I'm positive enough, boom, it fires. Okay? So it's like a giant calculator. Okay? You're adding up all these signals. And then it has to be right here, though, is where that signal has to be positive enough to fire. And you get your typical action potential with the voltage-gated sodium channel opening up. Okay? But you'll notice that this thing is wrapped. The axon, at least this type of axon, this is called a myelinated axon. Some axons in the nervous system are unmyelinated. They don't have this sheath around them. But this is a whole cell right here. Okay, this is called a Schwann cell. It's live, it has a nucleus and everything else. And it wraps around that axon just like this. Usually there's dozens of layers. Okay, and what it does is insulate that axon, but it also makes it impossible for sodium and potassium to cross the axon membrane. So you don't have an action potential right here. Okay. What happens, this is kind of interesting, on the myelinated ones, when the positive charge from the sodium rushes in, all right, basically what happens is, remember under the rest of the membrane it's negative, okay, that's the resting potential, it's negative. It's positive here, all this will be negative along these walls here. And that acts like an attractant, almost like a magnet. It pulls the negative charge towards the positive charge, but what's that going to do back here? Negative charge moving this way is going to leave positive charge back this way. So when that gets positive enough, at each one of these little nodes here, there are voltage-gated sodium channels. So they will dump in positive charge at that node, and then the same thing happens. Okay? So you get sort of this leap of the action potential going between node to node to node. Okay? And that happens to be faster. Okay? That happens to be faster than waiting for the next sodium channel to open. Okay? So the myelinated fibers here, the axons, fire faster. Okay? And the difference may be only a few milliseconds, but it makes a little difference in the way you sense things. You probably had the sensation where you put your hand on a hot stove, you're already pulling that off, okay, before you feel the pain. Right? You step on a tack, you're already shifting your weight before you feel the pain. Okay? That's because those fibers that feel the initial stick or burn, they are faster than the ones that carry the pain receptors, okay? The pain messages. Okay? So that's it's kind of interesting the way that works. But these are common in the peripheral nervous system like this. This would be like a motor neuron possibly in your spinal cord, sending an axon down to, say, the nerve maybe, okay? Because in the peripheral nervous system, everything but the brain and the spinal cord, okay, they have these Schwann cells like this, okay? And the phospholipid that forms that membrane, just like it's a phospholipid bilayer, just like all cells, okay? It has a special chemical in there, a special lipid called myelin. It's a little different than the phospholipid you've seen before. Okay? I'm not going to let you worry about what the structure is, but we'll just define it as a little different. The head is a little different, the hydrophilic part. Okay? And that's called myelin. Okay? And so that chemical gives this the name the myelin sheath. That's why they call it that. Okay? But it's still a Schwann cell. Okay? And the way you can look at this is just like rolling up a tube of toothpaste around a pencil. Okay? You roll it up, and then sooner or later you're going to get to the head of the toothpaste, and you're going to have some of that stuff that's stuck up near the cap. Okay? And that's exactly what they look like. You can see them all rolled up, but you'll see part of the cell with the nucleus in it on the outside in a little bulge. Okay? So that's how the Schwann cells do it. Inside the brain, okay, there's another type of cell that I'll come to here in a minute. You can see the names, called the oligodendrocyte. And it doesn't wrap itself around the axon totally like this. What it does is sit between the axon. It reaches out with, like dendrites almost, and wraps those arms around the axons. Okay? So it'll form a connective sheath right here, and you might have another oligodendrocyte over here that forms the next one. Okay? So they reach out, but they also give the brain a little bit of rigidity. So it's not just a big mass of jello. You know, it's not a gel. With all these connections, it gives it sort of a firm structure, about the consistency of a mushroom. You know, if you've ever sliced a mushroom for dinner, just that firmness is about how hard the brain is, the brain tissues. Okay? And you'll see that when we do the sheep brain. Okay? You'll be able to get a feel for that. Okay?
But this is a nerve muscle anywhere in the body?
It has to be peripheral. Okay? Outside the brain.
Okay.
Okay? Inside the brain, it's the other types of cells that do this. Okay? So whenever you see a Schwann cell like this, it's a peripheral nerve. Okay? Okay. And the nodes have a special name. I assume it was a French anatomist or physiologist that named these things. Ranvier, the node of Ranvier. That's the important part. That's where the voltage-gated sodium channels are here and here and here. So you get that, because it jumps, the charge sort of jumps. Saltare means to jump. They call it saltatory excitation or saltatory impulse, a jumping impulse. But like I say, it's faster than without it, than without this sheath. So, words on this page. Dendrites are new, okay? The axon hillock is new. Well, the axon itself is new, okay? The Schwann cell, also known as the myelin sheath, that's new to you now, okay? That's about all you need to get from this slide. Don't worry about things like axodendritic synapses and words like that. You know, you'll forget that in a month anyway. Node of Ranvier, yeah, you should have that in your word bank. Okay, we'll move on a little bit. Before we go into the types of nerve cells and this sort of thing, let's sort of break down the nervous system so you know what we're talking about when I say the central nervous system, the peripheral nervous system, and that sort of thing. Basically, the brain and spinal cord is the CNS, or the central nervous system. Everything else is the peripheral nervous system. Okay? And we can divide the peripheral nervous system into a couple of different systems. The somatic nervous system, this is what you normally think of as your nervous system, your sensation, you're controlling your muscles, things like that. Okay, this is sort of an unconscious nervous system that we have as part of the peripheral system. It's involuntary. This is what controls your heart rate. It controls the opening and closing of the smooth muscles in your capillaries. It controls the blood flow through your kidneys. It controls how many contractions you get in your stomach and your intestines. It controls bladder contractions and all sorts of things like that. It's sort of your unconscious internal system that works on all your organs. And that, called the autonomic nervous system, can be broken into two different parts. Okay? So we have one. This is normally known as the fight or flight system. This is called the sympathetic nervous system. Okay, it's all unconscious. You don't control releasing adrenaline or the hair standing up on your arms. You know, it's an unconscious internal system, but it's part of the fight or flight system. Okay, the parasympathetic system. This is the one I don't like just the relaxing responses because that's not really necessarily true. I call it the rest and digest system. Okay, now I'll tell you why here in a second. We're going to get into this in much more detail when we actually look at the anatomy of the system. But I call it the rest and digest because this is the system that is going to sort of be doing the opposite things that the sympathetic system is going to be doing, but it's not always converting to a resting state. This is actually excitatory when you're talking about increasing the stomach contractions. Okay, it's actually excitatory in that sense. Okay. Or increasing your intestinal contractions, things like that. So it does have motor parts to it that will make things work, okay, not just relax. Okay, so I say rest and digest. And we'll talk about the system when we come to it. But the hallmark is you can almost guess what's going to be what part of what system. When say you're running from that bear, okay? You're in your fight or flight response, okay, or you should be, right? Do you want to be salivating? Wasting fluids, right? You're not eating, you don't need it, you're wasting fluids, you want to, you know. So what system do you think actually stimulates the salivary glands to produce saliva?
The parasympathetic.
The parasympathetic, right. Yeah, so almost anything, it works every time. When you're running, do you want the capillaries and the blood flow and the airways, the smooth muscle that controls the airways in your lung, do you want them constricted or do you want them open? You want them open. You want to get a lot of blood to your lungs. You want to get a lot of air in your lungs. That's the sympathetic system that does that for you. Okay? So the parasympathetic system would actually close down the airways. Okay? And that's the kind of thing that you take inhalers for, to block this system from closing it down. Okay? That's why you take those inhalers. They're blocking the nerves and opening and relaxing the smooth muscle for asthma attacks, things like that. Okay? Do you want to be constricting your stomach and digesting your food and squirting digestive juices while you're running from the bear? No. So that's, you can see, that's part of your parasympathetic system that does that. Okay? Do you want a lot of blood flow to your fat, or do you want it to your muscles? That's exactly what happens when you stimulate the system with sympathetic adrenergic neurotransmitters. The blood flow, they actually have two different types of receptors, one that shuts down, ones that open up. So when you're in that fight or flight mode, the ones in your muscles, the blood vessels open up, and the ones in your fat close down. Okay, so you're shunting all the blood and oxygen to your muscles. Okay, and then it's just sort of the opposite is what the parasympathetic does. Okay, and it shuts it down in your GI system as well. You don't want to spend blood and oxygen digesting food, okay, while you're running. Okay, you want everything you can to get to your muscles ready for fight and flight. Okay, so like I said, you can almost guess. Just think, do I want my eyes dilated while I'm fighting and running, or do I want them constricted? Dilated, yeah. You want to get more light in there? Yeah. So it's the sympathetic system that dilates your pupils. Okay, and that's why some of the drugs that affect, say, the congestion in your sinuses. Right? That would be, if you're congested, okay, that's a parasympathetic kind of thing. You don't want to be wasting fluids in your sinuses when you're in a fight, okay? So you can tell that the parasympathetic is the one that stimulates you to make mucus, okay? So that's the one you want to block if you want to decongest in cold medicine, okay? But you can also see what it's going to do. This would tend to constrict your eyes. This one dilates, and parasympathetic constricts them. So, but you take a drug that blocks that, you're going to let your eyes dilate a little bit, and you're going to get a little blurred vision. Okay, which is why, if you look at the side effects on drugs like that, one of them is blurred vision. Okay, so you can almost predict what the side effects of these drugs are going to do depending on which one you're using or which one you're blocking. Okay, so and because this system uses norepinephrine and noradrenaline, which is the same thing, same chemical. Okay, they call it. It looks like adrenaline, so they call it the adrenergic system. Okay. It's also stimulated by adrenaline itself. Your the medulla in the what's the gland? Right above the kidney. Adrenal gland. Yeah, I'm having a stroke. The adrenal gland. Okay, that's what releases the adrenaline. All right, and that's part of this system. All right, so that just dumps it out into your bloodstream. Okay, so then it's everywhere, and it happens pretty quick. Think about how fast, when you hear a loud noise, how fast your hair stands up, and you feel like your blood pressure is going up. Okay, it's pretty quick. Okay, so it gets in your system real quick. But this system over here, the parasympathetic motor system, and the sensory system too, they use acetylcholine. Okay, they don't use adrenaline or noradrenaline like this. They use acetylcholine, so it's called the cholinergic system. Okay. So whenever you're taking cold medications, okay, you can either take stimulants that stimulate this system, and they will shut down that mucus production, okay, or you can take drugs that are anticholinergic, okay, and block this system, which will stop the mucus. So you can kind of do it both ways, okay. So, which is why if you use one that's adrenergic and you're stimulating the sympathetic system like phenylephrine, that's why a lot of people feel jittery when they take that, okay? Or they'll say it makes them feel nervous, okay? It can also cause blurred vision because it's, you know, it's opening the eye, it can dilate the eye, that sort of thing. Get too much light in, make you light sensitive, that sort of thing. So those are the kind of side effects you get with some of the cold medications. Okay? And then the ones that are actually trying to stimulate this system, you get other sorts of things, other sorts of problems. If this is making you produce the mucus, so you want to block it, okay? But if I block that, if I block the cholinergic system, do you think I'm going to get constipated, or do you think I'm going to have crampy diarrhea? Exactly. You guessed it. You're shutting down the parasympathetic system, so you're not stimulating the bowel, and one of the side effects, constipation. Okay? So that's how much sense it makes. Okay? When we get to it, we'll actually talk about the structure of the nervous system. So, some basic neuron types. Your book covers these. There are a lot more. Okay, there are dozens of different types of neurons in the brain, but these are some of the basic types that we need to just think about and have a basic vocabulary for. Okay, the pyramidal one looks. Usually they look like a little triangle. Okay, I think that's how they got the name pyramidal. But there are pyramidal tracks that look like little deltas in the brain too. That might also be how they got the name. But this is what they look like in the brain. Many dendrites. Okay. One axon sending the message somewhere else. Okay. Multipolar, if you consider these are the poles, so it's multipolar. This is what a typical motor neuron looks like that lives in your spine, okay, or in the brainstem in the case of the cranial nerves. Okay. The ones that the muscles of facial expression, those motor bodies of those motor neurons live in your brainstem. Okay. And let's see, and they're going to send out a message like this. Their axons can be mighty long, because if the body lives in the spinal cord, how am I going to move my big toe or the peroneus brevis? You know, if that axon actually is that long, it goes all the way down your leg. This thing is 1 meter long at least. Okay, so because the body, the nerves don't live outside the spinal cord. Okay. Typical sensory neuron looks sort of like this. The body actually lives outside the spinal cord, and I'll show you where it lives. But this would be in the periphery. Let's say this is some sort of pain fiber. This will create an action potential that will just move up the axon this way and keep on going until it stimulates an interneuron up here or goes up to the brain or whatever. Okay, so that's a typical sensory neuron. It has basically one pole, if you want to call it that way. This has multiple poles here. Bipolar, 2 poles. Okay, the signal actually goes through the body along the membrane of the cell and then out the other side. Okay, so that's interesting. And these you only see. I'll show you a couple pictures of these things, but you only see it with the special senses, sight, smell, and hearing, that sort of thing. So that's the basic neuron types that you should just have in your word vocabulary. Okay. Let me just show you some pictures of these things. Yeah.
You said the multipolar is in the brainstem?
Well, only for the motor nerves.
Okay.
You know, like the facial nerve that controls your facial expression, or the trigeminal that controls your chewing, that sort of thing.
Okay.
Those motor nerves are high up. They're cranial nerves, as opposed to the other nerves coming out of the spinal cord.
All right.
And we'll learn all 12 cranial nerves by the next 3 weeks. Okay. Just to show you some pictures of these things, different ways of staining, looking at them. Okay, this is another fluorescent stain here where you get them to glow. This is probably under ultraviolet light. They've got special glowing chemicals. You can see that these are pyramidal neurons. They have that little triangular sort of shape. But this also gives you a sense of a layering here. Okay, then you have axons going up and connections going between. Here's another layer here where they're more common. Okay. We actually have 6 layers of neurons in our cortex, okay, which is different than lower animals who only have 3. So at some point, and I'll have to check. I don't know if all mammals have 6 layers, but that was a big evolutionary step. You could call it a mutation, where you get like a gene duplication, where you get a doubling of the layer of the brain, and you have 6 layers of neurons instead of 3. Alligators, frogs have 3, okay? But we've got 6. And that was a big step for the computational power of the brain, okay? And here's another pyramidal one here, stained a little differently. This just happens to be the axon right there, because that's typically what they look like, okay? You get these big dendrites bringing in signals, okay? And then when that axon hillock right there gets positive enough, boom, it'll fire down that axon and then stimulate the next one.
So the axon is skinnier
Yeah,
than
yeah, typically it is. Yeah. You'll see a lot of them that look like this, like I say, that triangular shape. It tends to come out of the base right there. It's like it's coming out of the base of the triangle right there. Okay, that's just, I've seen, enough pictures of them. Just, I'm not an expert on it, but that just tends to be where they come out of the base. So, and every one of these little dotted areas here, these are all synapses. Every one of these little glowing things there, those are all synapses. So these things, there are a lot of axons and dendrites that you can't even see here. Okay? So somebody had to clear out the forest just so we could see a few of them. Okay? Here's another one, different way of looking at it. All these little things right here, these are all synapses. Okay? Some sort of fluorescent antibody stain for synaptic proteins or something. So thousands of connections on these dendrites, and then who knows, that might be the axon there on this one, but it's hard to tell. Or it might be that one right there, because you don't see too many synapses on that one. Nice thing. This isn't a great picture, but this gives you an idea of the layering of these things. Okay, not all the layers have the same number of neurons in them, but if you just remember 6, you just might get a little extra credit, all right?
So that means
6 layers. Here's one of the bipolar neurons. This is olfaction and sense of smell. You can see, here's one of the neurons. It's going to send one of its projections up here to these olfactory cells. Okay. And then it's going to send a projection down this way. And this actually becomes part of the olfactory nerve, okay, which is what's going up through the mucous membrane in the upper part of your sinuses. Okay? It's going up through the cribriform plate, and it's going to synapse with axons and nerve bodies up in the ganglion, the olfactory ganglion that sits right above the cribriform plate. Okay? Once you get in the brain, you don't call it a nerve anymore, you call it a tract. So the olfactory nerve is going up to the ganglion, and then from there on back into the brain is called the olfactory tract. Okay? I don't know if I've actually talked to this class at all about this, too much about the sense of smell. I think we did once when I talked about olfactory proteins. You got about 400 different kinds.
Some don't use anymore.
Sorry, and about probably 800 we don't use anymore. They've been mutated, and we don't. The cells don't make those proteins anymore. The genes are shut off. But about 400 of them, that's still kind of a lot. And you've got thousands of each one sitting up in your olfactory area there. But the way the sense of smell works, if I haven't already said it, obviously one molecule might come and fit in one of those receptors on one of those 400, okay? But it also might fit in number 37 and number 342 just by chance. It might have the right kind of size to fit into different ones. Well, what your brain does is add up those special types of, your brain can see the different signals coming from the different receptors, and it says, well, 27 and 42 and 399, that's bananas. See, that's what your brain does. It adds up those signals and interprets it as a smell. There's no such thing as the smell of bananas except what goes on in our head. Those molecules don't smell like bananas. Okay? As a matter of fact, they don't even know they have a smell. Okay? It's only when molecules float into your olfactory receptors and give you some sort of a pattern, okay, that your brain interprets as the smell of a rose, okay, or the smell of dog poop. And many times it's a mixture of chemicals, you can guess, okay, that's going to give you that real weird pattern, okay, which is why some people are really good at sort of pulling out those things and smelling the difference between, oak and willow in wine and that sort of thing, they've got a really good nose and they can pull out those different pathways even when they're all mixed together. Okay? Some people are good at that and others aren't. I'm not, okay.
So most people then have pretty much the same receptors, since we all kind
Yeah.
of smell.
Oh yeah, we pretty much all have the same 400 receptors. But some people are probably just born with better connections, and they can smell these differences better, okay? And there are some people that are actually born without certain receptors because they can't smell certain things. And the psychologists have tested this, physiologists have looked at this. Some people really can't smell certain things, okay? So we know they don't have those receptors, okay? And that's genetic. So there's a little bit of difference between individuals. So, okay. Here's another bipolar neuron in our retina, okay? These are called the rods and cones. This is what it looks like under the electron microscope. These are the rods, here's the cones, okay? This is for color vision, this is for black and white, gray vision, and mainly nighttime vision as well. That's why you don't see much color at night, because these things need a little bit more power to fire, a little bit more light to fire. But these are more sensitive, so you use these to see at night, okay?
Those are all nerves.
These are all nerve, well, yeah, you could consider them. They're photoreceptors in the retina of the eye.
Okay.
Okay, but that's how they line up like that. It's almost like a carpet. Okay,
Yeah.
this is in the back of your eye on the retina. Okay, the weird thing is the light comes from this direction. It doesn't land on them this way.
Right.
The way our retinas develop in embryology, they're turned inside out. So the light has to actually come through all this stuff, and then hit one of the receptors here. Okay. Other animals have better designed eyes than we do, like I think squid and octopus. It's arranged the right way, so the light's coming in this way. Okay, so they have interesting eyes. Okay. But here's the bipolar ones. It's going to send the signal right through here to something called a ganglion cell, and we'll go over this big time when we talk about the eye, so don't worry too much about it today. But this ganglion cell right here, that axon is what's going to send the message back to the brain and the optic nerve.
Now, when somebody has eye surgery, does it affect that in any way?
Well, you can, yeah. You can rip these off the retina or break these connections, yeah. I'll tell you my experiences with my eyes when we get to the eyes, I guess. So I won't go through it right now, but I've got a few blind spots that aren't natural, let's put it that way. Everybody know what their blind spot is? Yeah. How do you find it? Not too far out there, no. That would be your field of vision. You know, when your hand disappears, that's your field of vision. Okay, and it's not quite 90, it's a little more than that, but I can sense things back here behind me a little bit. But your blind spot is if you close one eye and say, focus, put your finger right up and cover this spot right here. Okay? All right? If you're closing your left eye, okay? All right, put your finger right there. Keep staring at this spot, and when you move your finger over here, probably about 6 inches for you, the end of your finger is going to disappear.
You're supposed to stare at it.
I can still see mine.
No, not your finger. Keep staring at this spot, and just move your finger over here.
I can still see mine.
Well, yeah, you're way too far. It's if I do it, look at me. So I'm looking at the tip of my finger and I'm looking at that electrical box. I keep staring at the box and I move my finger about 6 inches and boom, it disappears.
Oh, so keep looking at the spot.
Yeah, keep looking at the spot and just move your finger away.
With the left eye
Don't
closed.
do it too fast because it's only about 6 inches. It's not out here.
Does it change the hands whenever you do different eyes? Or?
Yeah. Yeah, you
Okay,
gotta go
so
the other
you-
way.
Okay, so, okay.
Just don't follow your
So
finger.
if I'm closing my right, I can go that way.
Yeah.
So for the left eye's closed, I move my finger to the right.
Yeah. You keep your left eye closed
Right.
and move your finger to the
Oh,
right.
that's not on the tip.
Wiggling it in and out of the
I don't lose my finger.
It'll go in and out of it because you'll pass it. There's the blind spot and all of a sudden
Oh,
it comes
okay.
back.
Oh, maybe I did too fast then.
It cuts
The other
off,
way.
cuts off the top of your finger.
You gotta go to the
I
right.
don't.
Oh, I did.
You close one
Your
eye.
blind spot is on the outside of your vision, not on the
I mean, I'm
inside.
sure it's there, I'm just not seeing it.
Well, anyway,
Well, sorry, I'm seeing it.
practice that at home, we'll put that on the test. Extra credit next time. The reason that happens is because the light that's falling on your optic nerve, it has no photoreceptors, so there's a dark spot there. So anything in your field of vision that falls on the optic nerve is not going to be perceived. Okay? So that's what causes the blind spot. So where all these nerves are collecting and going to the back of the eye and then diving into the optic nerve. The light that falls right on that spot, you can't see it, because there's nothing to see. Okay? That's why your finger will disappear.
So the green is the back of the eye, and that's the front, or no, that's the back,
Well,
and
this
that's
is the
the
back
front.
wall.
That's
This
the back
this
wall.
is the front part that you think of as your retina. You've probably seen a picture of your retina. What you're looking at is the ganglion cells right here. Okay. The reason it's sort of reddish is because of the capillaries and the blood flow. Okay.
So confused.
But right now, all it is an example of a bipolar nerve. Okay. Don't worry about facts and figures and other things and blind spots. Yeah. We'll get to that later. Here's another bipolar neuron. It has extensions that go up to this little organ here in your ear. When these membranes vibrate, you'll be naming all these membranes for me by the time we're done here. Okay. When this thing shakes little hair cells here, it fires. Okay. Here's the body of the bipolar neurons, and then they go back into the acoustic nerve here, the cochlear nerve, and go back to the brain. Okay. So the bipolar cells live in this whole thing called the cochlea.
And when we go over the ear, this will make a lot more sense to us, this
Oh yeah,
picture.
oh yeah,
Okay.
you're gonna know this by heart.
Okay.
Because this could be a typical slide for the final.
Okay.
All right. Here's two nerves touching each other. Here's the synapse. Okay. Which direction is the signal going?
Where is the
Down.
Why up?
Wait a second,
Because all those things are up there.
Well, they're up there, but what are they full of?
The
Color.
natural.
See if I'll-
Full of color? Full of purple color. These are the vesicles that are going to be releasing the chemicals into the synapse.
Right.
So the message is being sent from this one to this one.
Oh,
Okay.
Okay.
This is the vesicles with the neurotransmitter. This could be acetylcholine. Okay. So it's going to be dumping that in here. And here's where the channels are, the chemically gated sodium channels that are going to, fire on the next one. Yes.
But I'm just, so there are two nerve axons, one on the left, one on the right?
Well, I guess it could be an axon. It's more likely the body of another nerve here.
Or the button end,
Or an
is
dendrite.
that, we're looking at the two button ends? I guess I'm just very confused.
Well, yeah, this is highly enlarged. So all you're
Okay.
seeing is just the synapse, really.
Okay,
So this is the end of an axon.
Okay.
And we know that because it's going to be sending a signal out.
okay,
And this could be a dendrite wall, this could be this wall of the cell body or something. We don't know what it is.
Okay.
But you see there's no vesicles here. So,
Right.
So it's not going to be sending anything up. Okay? The neurotransmitter has to be collecting in these things in the vesicles.
and how do you know it's the end of the axon when you look at it, because that's where the vesicles are?
Yeah, sure.
Okay,
That's the only place that the signal will be sent.
okay.
And this is more PBS, I guess, than anything else, because you can see how complicated it is, and that's my whole point. I just want to show you how complicated it is. Remember we talked, though, about the end of the nerve where calcium had to come in, okay? The voltage-gated calcium channels were letting calcium come in. The reason is because right here, calcium is the trigger for this apparatus right here to pull the string and open these two membranes and dump out the vesicles. Okay? It's an electrostatic charge effect that causes a change in the shape of the protein, and that causes a mechanical effect, okay, which pulls this thing open. But the vesicle won't even land on the wall here unless it has the right receptors here, almost like antibodies. When these things match up with these here, it forms this complex, okay? And that complex is now waiting for calcium. Okay? As soon as calcium comes in and lands on these things, I think it's the synaptobrevin that it binds to here, that'll cause all these proteins to just go and just pull it open. Okay? And then they dump out the neurotransmitter. Okay? So that's and you should also be thinking in your head, these are all proteins, several of them. Okay? Every one of these is made by a gene. Okay? In your DNA. Every one of these could have a mutation in the gene and it won't do its job right. Okay? So every one of these proteins could be the basis behind a neurological disease. Okay? So that's how you need to be thinking when you think proteins. Yeah?
Is that also why some people have a faster reaction time?
Yeah, it could
Or
be.
slower reaction?
It could be, yeah, yeah. And think, this is all going on in the brain too. You know, when these things are, you know, so it can affect thought processes, you know, you know, I'm not an expert on it. Who knows? I'll plead ignorance. What is the real basic cause of something like schizophrenia, where the wiring goes wrong and you have delusional thoughts and things like that? Is it something like this in the wrong place of the brain, you know, where most people are rational? Well, it could be, at least in my mind, okay, but I don't know. Okay.
What would be the reason, like, what I'm trying to follow through this whole process is like Alzheimer's, let's say. It takes so long, and then all of a sudden, you know, they start exhibiting signs of Alzheimer's. What's actually happening is it mutation just finally maturing, or?
Well, there are some genetic causes for Alzheimer's, and they're
Right.
related to hyperlipidemia, high cholesterol, lipid levels, things like that. But, you know, I'd have to do some reading to give you an exact answer, but it's a buildup of certain proteins on the neuron wall. Then it causes a breakdown of the nerve bodies in the axons. So you're actually losing nerve mass. Okay, you're losing brain mass in Alzheimer's. You take an MRI or a CT scan of the brain, you can actually see that it's shrunk. Okay? And it has areas that are just sort of not there. Okay? So something is being toxic to the cells. I don't think it's a problem with transmission per se, but you're killing the neurons themselves. Okay? So, but I'm sure in the last couple of years since I did any reading on it, you know, it's more is known about it, and I'd have to look at that to really give you a good description. This just happens. Here's your vesicles. The stuff has to get back into the vesicles. Okay? And this is just one experiment that they did where they took dopamine and serotonin. These are chemicals that are widely used in the brain. And they took something that had a similar sort of structure to it, at least it fits in the same receptors, the same pumps, that pump it back into the vesicles. And this is sort of a glow-in-the-dark molecule, okay? And you can see that it's being pumped back into this vesicle. Okay? So that's what happens. There are special membrane pumps that take these things and get them back into the vesicles.
Now, does that require ATP to pump that back
Yeah,
in?
yeah, they're pumps. Because
Okay.
you're pumping against the concentration gradient.
So it would be the ATP.
Yeah, yeah. Which is why the mitochondria have to be down in the axon too.
Okay.
Creating all that ATP. Here's a realistic look of that axon with the myelin sheath. Okay. This is coming out at you, and it's cut off, transected like that. So there's a mitochondria right inside the axon. Okay. And these are probably microtubules that are coming at you too. They've been cut off as well. But look at a couple of dozen layers of that Schwann cell is wrapping around there, and then here's all the toothpaste up at the end of the tube. Right, here's the nucleus. Okay, so this whole thing is the Schwann cell. This is the cytoplasm of the Schwann cell. But all it is membranes down here. There's no protoplasm in here. It's just membrane on top of membrane on top of membrane. So there's no transmission. There's no action potential that can get through that. Okay. And like I said, just like this one says here, it's the oligodendrocyte that does this in the brain. The Schwann cell does it in the peripheral nervous system. And you'll be happy to know you've already learned the epi, peri, and endo. So now we just need to apply it to the nerve structure of the peripheral nerves. Okay? Around the whole nerve, okay, is the epinerve, just like the epimysium in the muscle. Okay? Around the fascicle here is the perineurium, just like the perimysium in the muscle. Okay? Around each nerve cell is the endoneurium, okay? Just like the endomysium in the muscle. Okay?
So your nerves are essentially designed very similarly to your muscles with,
In this sense, yeah.
in that sense, okay.
Yeah. Yeah. Bundles on top of bundles, and then they just have different connective tissue layers.
Okay.
You can see these like this, where you can, you take a fascicle like this, and a fascicle like this. That would be the perimysium, or perineurium, I'm sorry, perineurium around here. Perineurium around these fascicles. And somewhere out here is the epineurium surrounding the whole nerve. Okay, this is probably just fat tissue inside the nerve.
There's fat tissue inside the nerve as well?
Can't be sure. Connective tissue. The outside Now here's a blown up part of this. Here's your node of Ranvier. This is where two Schwann cells are touching each other. Okay? But you can see this light blue stain. That's the endoneurium. Very thin, very delicate. Okay? And so put a bunch of nerves together and you get a fascicle. So this would be perineurium here, around the fascicle. And somewhere way out here around the muscle would be the epineurium. Okay? So, but that's interesting. They got a shot right there. I think I have another one right here showing a node of Ranvier. This is where the sodium channels are going to be that are going to continue that action potential. And same sort of thing right there. And this is something we need to go over. It's interesting how the brain is sort of put together with different types of supporting neural structures. The neurons, like I said, there are about 100 billion of them, but they're sort of protected by all these other different types of cells here. And there's three of them we're going to talk about here. This is that oligodendrocyte I was telling you. It has little arms that go out and wrap around the axon to form those sheaths, to form the myelin sheath. Okay? But you can see it's got several arms, and it's actually working on two different axons. Okay? So it doesn't wrap the whole body around there. It takes an extension and then wraps that around, and then another oligodendrocyte may continue the process as it goes on down. Okay? So this serves, like I was saying, to give the brain a little bit of rigidity, a little bit of structure. It's not just a bunch of neurons like Jell-O laying together. Okay? So that forms part of the rigidity of the brain, like I said. And that's pretty much all their function is, as far as I know. Okay? The astrocytes are interesting here. They have little arms that come out, little extensions that come out, and form like little foot pads that wrap around the capillaries of the brain. This is the blood flow that's going to be giving nutrients and oxygen to this neuron. Okay? There's a lot of blood flow in the brain. A lot of blood flow. Your brain is using, 10-15% of your oxygen supplies at any one moment. And probably the blood sugar too, because it's firing and using this stuff up. But you see these little foot pads, they wrap around that capillary endothelial cell. So whatever's in there, and you can see how small it is, that's one blood cell right there. Okay? Whatever's in there can get through the endothelium, maybe, okay? But can it get through these foot pads? Okay? They call this the blood-brain barrier because there's nothing else like this in the body. Okay? Your brain protects itself from certain toxic chemicals, and your kidney is doing its very best to get rid of these things. Okay? And even some drugs that you might want to get into the brain can't get in. Okay? Like we talked before about the first pass effect. If you take a drug orally, it goes through the intestine, it gets absorbed, it has to go through the liver before it can get out into the body to do anything. If your liver detoxifies it and kills the effect of the antibiotic, it's not going to work orally. Okay? And that's why some antibiotics can't be taken as pills. They have to be given in shots or IV. Okay? But even then, you can inject things into your veins, and it'll go all the way around the body. It'll sail in through these capillaries, but the astrocytes may not let it into the brain. Okay, so certain antibiotics are not good for brain infections, for meningitis, things like that. Okay, you have to have special. Usually, you modify them a little bit to make them more fat soluble chemically. Okay, like you put a lipid on it or something like that. Then it has a better chance of getting through. Okay. But the astrocyte is what is creating that term. It's called the blood-brain barrier. Okay? So just have that in your vocabulary to understand what that is. But these are also responsible, like I said, letting anything through. These cells are responsible for the sodium, potassium, calcium levels around the neuron. So you got to keep those levels right, or the action potentials don't work, right? Okay. So it's not the cell here that's really keeping all those levels right outside the cell. It's the astrocyte. Okay, it's the astrocyte that's in control of that. And let's see, the last two here, the ependymal cells. Pretty much you can be just satisfied with knowing that they line the cavities, the fluid cavities inside the brain that we'll be looking at. Okay, and they produce the cerebrospinal fluid. Okay? It oozes out from them, and they're the ones that are creating the fluid. They also have little cilia-like projections on here that creates a movement in the fluid. So the fluid is actually being sort of paddled around your head, or paddled through these spaces. Okay? And so, but that's pretty much all they do, and that's all I require you to know about it.
Produces what again?
The cerebrospinal fluid, the fluid that's around the brain and inside the ventricles in the brain. And you'll understand what we mean by that when we get to the actual brain anatomy. Okay. The last one can be looked at as, I don't know if we've talked about the term phagocyte. Okay, that's a cell like some of the white blood cells that will engulf bacteria and dead debris, things like this, and destroy it and eat it and dissolve it and get rid of it and that sort of thing. Well, that's the goal of the microglial cells. They're sort of the garbage men in the brain. Okay? They are mobile. They can move around. Okay, and they'll take care of things. So, like little amoebas crawling around. Okay.
The cleaners.
Yeah, the cleaners. They take care of the garbage and trash and the dead tissues and cells and things like that. Okay. And, okay. Is there anything else I want to say about this? Oh, well, like the slide mentions here, you're pretty much born. There may be some slight exceptions that they've found recently, but you're pretty much born with most of the neurons you're going to have all your life. Your brain actually just gets bigger because it's sort of expanding. Okay, it's and growing more connections. Okay, but what that means is these aren't dividing. Okay? They don't go through mitosis. Okay, which means there's much less likely that they don't even have a mechanism to do it. So it's much less likely that they would become cancerous because they don't know how to divide. Okay, so most brain tumors come from these other types of cells. That's why you'll hear words like ependymomas, gliomas, oligodendrocytomas.
So astrocytes is the one that.
Yeah, astrocytoma. Yeah, that's a cancer of the astrocytes. Okay, so those are the kind of words you'll hear a lot. Okay, and sometimes they just call them gliomas, but because neurons don't divide, they're much less likely to actually have a problem with division and divide too much. Okay, so that's the issue there.
So once you kill them, that's it.
Yeah, pretty much. Yeah, you kill brain cells and they're dead. They're gone. Okay. Sometimes you can rewire the brain, but that's called plasticity. But that gets harder and harder to do as you get older. Okay. Kids can actually do it fairly well in young infants. If you have brain damage at that stage, it's really interesting how different parts of the brain can rewire and take over the activity that part of the brain used to do. Okay, so you might have a part of the brain that was damaged, which affects hearing. Well, then it rewires and puts the hearing center somewhere else. Okay, adults really can't do that. Okay? Not very well.
I might not be remembering this correctly, but when I was reading on the brain before, if I remember correctly, isn't that where also stem cell research comes into play through the plasticity?
Sure.
Okay.
Well, that's the whole point. You're trying to put something there that you can't make these cells divide and repair themselves. Okay? So you're trying to use stem cells and turn them into the right kind of neural cells that you want.
Okay.
Okay. And to do that, you have to start with a cell that doesn't know what it is.
Which is
what itself is.
okay.
So you give it the signals and turn it into a brain cell.
Okay.
So, but even then, wiring is the issue. And
Okay.
I don't know how they're really going to get these things to wire themselves up right, when most of this has to be done in early infancy and childhood, or it never works. Okay? I don't know if you knew that if you just blindfold a kitten or a dog or whatever, this is the studies that I've seen, for the first 3 months of their life, okay, the brain will not wire the optic nerve to the occipital lobe, and you just take the blindfold off and they're blind the rest of their lives. Okay? That's what I mean by, you don't start with everything you're going to have. You have to have this growth and connection as your brain grows, which is why people and kids who aren't stimulated get these weird personality defects and sensory defects because they didn't develop the right part of the brain to appreciate the activity. Okay? Language, if you don't learn language by the time about puberty, about 12, you never will. Okay? You'll never develop the verbal centers and the grammar centers in the brain that allow you to talk fluently. You might be able just to mutter some sounds and words and things, but you can't make sentences very well. Okay? So, and a few feral children that have been locked in closets. We know that's true because that's what happened. They don't know how to talk and you can't teach them. So anyway, the brain is a funny thing like that. So, on the other hand, you've got kids that have had the whole half of their brain removed for some sort of reason, whether it's seizures or tumors or whatever, and they're young enough that that brain rewires itself, and now they're walking again like normal when they don't even have the right part of the brain to, work the arm or the leg. Okay, it rewired itself somewhere else. So that's really interesting stuff.
Is this from like, I saw a guy had like an accident and he had brain damage on one side of his brain. And he had problems doing some kind of like everyday things, but then he turned out to be like, he could do like magnificent artwork.
Yeah, there's several syndromes like that I've heard of. It's almost like becoming an idiot savant, like an autistic. You damage a part of the brain, and we don't know why this is, but normal people tend to have a lot of inhibitory impulses that sort of, for lack of a better word, inhibit your creativity. Okay? And if you damage the part of the brain that's inhibiting that, you sort of set it free. These people become great artists and things like that, and that's well known. But it's rare, but it's well known as fact. And so, but it's almost like an autistic person who's a wonderful artist. Part of their brain has been damaged, but this somehow frees this creative power to come out.
How it is damaged?
I'm sorry?
How is it damaged on a autistic or Asperger child?
Well, the wiring is going off.
Is it born with it or something?
A lot of it's probably genetic, yeah. And there may be some environmental influences to it, but most of it's probably genetic. I mean, it's certainly a higher incidence of autistic kids in families. Okay. But they're still trying to find the right genes. Okay. That's the whole big thing is now to try to find them and turn them off. Or it's test formed in utero or something. But I think it's pretty well known that it's a lot of genetics now that causes that autism. But somehow the wiring is off, and yet you can have autistic kids that are just brilliant at math or something like that. And we don't know how those connections really work. There was a lot of study in that. So, you remember Rain Man? Autistic, and yet he had a fantastic memory, just unbelievable. Okay? Most people, that would be a hindrance. Okay? You really don't want to tie up all your neurons memorizing trivial stuff like phone books. Okay? That apparently, I think, would screw up most normal people. Okay? And so that's why your brain just says, no, you don't need that information. Okay? So, and most of us have a certain power of memorizing things, but what happens? Is there some sort of brain lesion that could turn that on and make you a super memorizer? I don't know. I haven't actually heard of that happening. Has anybody taken psychology and heard of that or something?
Well, what about people that have like photographic memory?
Well, that's the same kind of thing. Yeah, but they're not always have deficits in other ways. And so I just don't know what turns on the memory like that. There are a lot of theories.